Good morning. I am Amy Herring, a member of the Planning Committee for this webinar series. I'd like to welcome everyone to our webinar today, which is the last of a series of 4 webinars conducted by the National Academies of Sciences, Engineering, and Medicine. I would like first to thank the NIH Office of Dietary Supplements, and in particular, Drs. Nancy Pottishman and Jamie Gachet for sponsoring this webinar series. We are also grateful for the sponsorship of the Academy of Nutrition and Dietetics, and welcome Dr. Allison Steiber of the Academy to today's webinar. I would like to thank and introduce you to my fellow Planning Committee members. Please note that they will be moderating the discussions and asking questions during the Q&A. This is webinar number 4, and it focuses on innovations and special considerations when assessing dietary intake in pregnant women and children from the ages of 2 through 11. You can find more information, including agendas, the biographies of all the speakers, and the recordings of the prior webinars on the National Academy's website. The prior workshops address methods for assessing intake in the populations of interest, the frequency of data collection, and challenges in capturing an accurate picture of dietary intake. While assessment of intake from foods, beverages, and supplements is challenging at any age, this is especially challenging during pregnancy and early childhood. We know there can be changes in dietary consumption from pre-pregnancy to pregnancy and during the pregnancy itself. In the first webinar, for example, Anna Maria shared her work showing an average increase across the course of pregnancy in consumption of dairy and in consumption of protein among vegetarians. In addition, women report a decrease in pregnancy and consumption of caffeine and alcohol. Though we may worry some of this reporting is influenced by social desirability bias. Pregnancy complications such as nausea and vomiting or decreased appetite due to size of the baby in the 3rd trimester can also impact intake, and women with greater financial flexibility may be more likely to make changes. Thus, determination of how frequently to assess intake in pregnancy and when to do so can be a challenge. Now, because children aged 2 to 11 are still developing the cognitive capacity to report their own intake, multiple surrogate reporters, including parents and childcare workers, often provide information about the child's intake. These informants may vary in their ability to report on the actual amount consumed. For example, here you see my son Peter at age 2, disagreeing with a mallard duck about the amount of white bread each will consume. I imagine his intake of bread that day was considerably greater than I might have expected. And even state-of-the-art photography or video-based methods may have faced challenges in accurate estimation of his dietary consumption. Also, we know that nutrient intake has high variability over time in this age group, raising the question, again, of how often we should attempt to measure. In addition, older children may report, either over-report or typically under-report, with a degree of bias depending on child-specific factors, such as age and weight status. These and other numerous potential biases, use of different informants, and even instruments to capture intake, and multiple measurements across time complicate decisions about study design and provide exciting challenges for analysts, statisticians, and data scientists after the data have been attained. Combining the results of multiple studies in a systematic review or meta-analysis is an even more daunting task, given that studies of interest may use a variety of assessment approaches and may tailor them to suit their own populations. Use of sophisticated statistical models can help address biases associated with imperfect assessment, especially when certain types of additional information, often involving significant participant burden, are available. However, statistical remedies that occur after data collection is complete are no substitute for perfect intake data if we could get it. Today, we address a variety of areas that are important and interesting, including exploration of emerging, potentially more objective technologies and new measurement approaches with potential to address the drawbacks of standard techniques. Do current feasibility and validity warrant widespread adoption at this time? We'll look at key considerations for use of assessment techniques, including access, culture and setting, diversity, privacy, expense, and burden. We'll consider principles guiding harmonization of assessment across studies and across different populations. For example, can we get an overall picture nationwide of nutritional status of pregnant women and children aged 2 to 11? And finally, we'll spend time pondering the central role of the research question in guiding our selection of assessment and analysis methods. A few words now about housekeeping and our agenda. The first speaker after me will talk about various considerations when designing studies. The next three speakers will describe some emerging methods that might be used to assess diet, and importantly, how to harmonize data across different sources and different populations. Finally, our fifth speaker will explain how to choose appropriate methods. We will have time for questions, and the webinar will end with a panel of experts that include some of the panelists from previous seminars as well as others. We'll wrap up with highlights from our webinar series. Please note that the first Q&A will be from panel members and study sponsors only. Now, if you're in the audience and have a question, please use the Q&A button to send your question and which speaker to direct it to. They'll be read by me during the final panel time permitting. Also, please note that we will strictly keep to the times assigned for each speaker. Now, today, we've invited today's speakers to address important considerations cutting across pregnancy and the childhood age range of interest. Dr. Carmen Perez-Rodrigo of University of the Basque Country will discuss design considerations to minimize misreporting. She will focus on the importance of consideration of specific features of a group that can influence reporting about intake behaviors, noting the importance of culture-sensible food lists and composition databases, among other important considerations. Dr. Carol Boucher from the University of Hawaii Cancer Center will present some emerging technologies and their potential for increasing data accuracy and decreasing participant burden. Dr. Katie Loth from University of Minnesota will discuss the potential for ecological momentary assessment in improving estimates of dietary intake. And Dr. Diane Catelier of RTI will tackle the important question of harmonization of different methods for dietary assessment. We will conclude presentations with Dr. Angela Liza of the University of South Carolina, who will emphasize the importance of considering the research question when choosing an appropriate assessment method and planning any subsequent statistical analysis. Now I'd like to turn the microphone over to Dr. Carmen Perez-Rodrigo.
Thank you. First of all, I would like to thank the organizers for counting on me for today's webinar, where I will discuss issues related to health literacy, diversity, access, and other considerations when developing and implementing methods for dietary assessment. And as has been mentioned in some other of the previous webinars on this issue, the level of the information required when we assess dietary intake in children and in pregnant women will depend on the purpose of the study itself, whether it is for surveillance purposes, whether it is a research study, or whatever the purpose we might be willing or needing to assess dietary intake. And then the level of accuracy that we might be needing will be very highly variable. Then the target group, the population where we want to collect the information from, might be quite diverse. If it is for surveillance purposes, we will be willing to collect the same level of information from different population groups, and we must be sure that the quality of the dietary intake data that we are collecting is of the same level for all the groups. And then we will be collecting information in different contexts, and this will be challenging sometimes. Then the resources available that we have for that purpose will be limited most often, and this will restrict the kind of methods or technologies we might be willing to incorporate. So overall, it's a balance that we have to make when we make decisions about the method that we want to use to collect data about dietary intake, the level of the information we want to use, and the population, the groups that we are working with. So when we're talking about dietary intake in children, we have to bear in mind that there are developmental stage so their cognitive abilities and literacy skills will be varying depending on their age. Children have memory capacities that develop with age, and the concept of time and their ability to report on portion size is very limited, as well as when if we want to use methods based on frequency of consumption, because their ability for young children is very difficult that they can report the usual frequency of consumption for certain foods. And they will need the aid of their parents or caregivers for reporting for them. Food literacy can be a limiting factor as well, as children have can have a limited vocabulary for naming foods, and they may be familiar with a limited repertoire of foods. And also they have difficulties when trying to identify main ingredients in mixed dishes. So this is a drawback when they have to report on what they have eaten. So it's often that we need to ask other people, like their parents, tutors, caregivers, or maybe have joint reports and see how the report for children and the surrogate reporters match and how well they can be combined. Social disability issues has also already been mentioned in other presentations. And despite social disability issues might be less of a problem when we're talking about children's dietary intake, this has been identified as well, and particularly with older children. And again, we have to face the problem that we do not have only children reporting about their diet, but also their parents or caretakers reporting about children's diets. And then social disability can become an issue. Children's dietary habits vary across age. While younger children eat most of the food at home, then as they age, they incorporate more eating occasions out of control of their families. So it's difficult for the parents to report on what they eat. And other considerations such as weight status, body image, or restrictive image can also influence their ability to report. In addition, when we are asking children to report on what they are eating, we have to capture their attention and engage them in the reporting activity for several minutes, and this can be boring for them. So we need to add motivation to the interview so that we keep them going. So misreporting is a problem when we are talking about children's reporting of what they eat, and it can be biased towards reporting below usual intake or over-reporting over what they eat. Underreporting is more frequent than over-reporting, and it varies. And when analyzing misreporting, we have to consider who is misreporting or is more likely to misreport, what kind of foods children misreport more often, and why they do misreport. So there are differences regarding age, and it's all as children. Older children are more likely to underreport. There is not such clear difference regarding gender, and regarding socioeconomic level, lower socioeconomic, lower educated families, children from lower educated families tend to underreport or misreport. Area of residence is conflicting, and regarding ethnicity, there are conflicting results. There is also an influence of BMI or BMI or other anthropometric indicators that can influence misreporting. But then there is a confounding factor between ethnicity and BMI, since some ethnic minorities, the prevalence of overweight can be higher than in others. Regarding what kind of foods, it's more likely that children misreport on the kind and amounts of foods they eat outside the family context, and those foods that are eaten as snacks. And in children, over-reporting has been reported to be higher than in adults, and children try to when they are trying to report on what they eat, sometimes they introduce foods and report foods they have not really eaten. So it's crucial that we consider methodological issues that can avoid or minimize misreported by helping children and probing them to report about during the interview. And we need to more accurate descriptions of the food and try to minimize the subject burden so as to avoid low response rates. So it's crucial that we have qualitative research in regards of misreporting and who is involved and why this is so. So some of the issues relevant to consider when collecting data about food intake is diversity. Diversity in terms of the kind of people who is participating in the survey and the access to food and to a variety of foods is different depending on different population subgroups. We have to consider cultural issues and food literacy. Food literacy in terms of own culture, since food literacy is highly contextual. So it's of utmost important that when we use methods which are based on a food list, we include appropriate lists and recipes within the food list, and that the food composition databases are able to reflect the food composition information of these foods that have been added. Now, another issue is that different people may name the same foods with using different names, or even the names of the recipes can be the same, but the ingredients be different. So this is something to consider, and both on the side of the people who are in charge of collecting, retrieving the information, and when designing the dietary assessment methods. It has been suggested that adding the collection of labels, packages, and wrappers can help to improve the accuracy of food reporting in both food records and 24-hour recalls. It can be more difficult when using food frequency questionnaires, and then it can be useful to leave open-ended items so that people can add the names of relevant foods as suitable. Language can be an issue, and literacy and numeracy can also be issues when conducting dietary assessment surveys. When we want to have a random representative sample of the population, we want all the population subgroups to be represented at the same level. And this means that overwhelming burden on the subjects may undermine participation, and literacy problems may undermine participation. So it's we have to foresee, it's needed to foresee what kind of aids and incentives can be planned to encourage participation. 24-hour recalls have been reported as the method which poses less burden to participants. And in some projects, like for instance, in the context of the Spanish Perseo project, which was a school-based, community-based program, we try to, from the community-based approach and a participatory appraisal, social workers and teachers of schools were involved in helping parents to report what their children were eating outside the school. So by means of this participatory appraisal, we were able to support, we managed to support lower income and ethnic minorities involved in the project. It has been suggested that new technologies, particularly taking pictures as additional resources to help in food records and 24-hour recalls, can be useful and encourage parent aid in participation in large surveys for lower income groups. So that currently everyone can take photos of what they eat, and even outside the home or inside the home, they can take a picture of what they are eating and add and instantly send it to the server so that the food eaten is incorporated.
Carmen, I'm sorry to have to interrupt, but
okay.
we do have to move to the next presentation of Dr. Boucher. We can come back to this in the discussion.
Okay, thank you. So then I will pass on to Carol Boucher at the University of Hawaii Cancer Center. Thank you.
Thanks so much, by the way. Carol Boucher here, and looking forward to this. So just as great entry there, a variety of dietary assessment approaches assist with capturing the multi-dimensionality and dynamism of food consumption among women during pregnancy and in middle childhood and early adolescence. A challenge regarding dietary data collection among this group is navigating whether the child can independently report or record dietary intakes, or use a parent proxy with the child assisting, or the child report with the parents assist. There are different approaches to use image-based methods of dietary assessment. Broadly, these methods can be considered as passive, which is represented here as a wearable camera. Lots of images and data are collected. Users do not necessarily need to be engaged. For the active approach, a mobile phone can be used to capture focused images. The users are engaged and the images are of better quality. To put into perspective, about one image every 5 seconds generates lots of images which need to be screened, whereas the active approach uses focused images, about 6 to 12 before and after pictures a day. For the first pass, the passive approach may look something like this. Time elapsed 24 minutes. For the active approach, time elapsed 24 hours and 8 eating occasions. The second or after image of each eating occasion is not shown in this image. For the passive collection, about 35 images start to show the before and after. For the active method, the before and after are available for recognition, analysis, and amounts consumed. Some of you may have noted the colorful checkerboard square or fiducial marker. This slide helps explain the fiducial marker's value. Depending upon lighting and other influences, colors will change. However, to identify a food, natural color is one of the most important indicators, and the standardized colors on the checkerboard square allows a classifier to designate the original native color. So now that we have gone through some of the basics of the methods, let's look at a few approaches designed for pregnant women. This image-based app uses an active approach and the audio as supporting voice description and date and title of record. There's a fiducial marker. For this study, the image-based dietary assessment was compared to results from 24-hour dietary recalls. The infrastructure of this system includes an image and voice file, and analysis with reference foods and shapes. Also, structured follow-up phone calls are used if further clarification is needed. This image-based assessment was compared to 24-hour dietary recalls among pregnant women and found to have an acceptable relative validity. Here is a different example of using images. This study used Smart Intake, which also uses an active approach. And in this case, the biomarker, doubly labeled water, was used. Unexpected, the energy intake estimated from the app only reached 63.4% of energy from the doubly labeled water. Unique to this study, all participants were within an obese category. Possible explanations of this. The PIs observed that the participation was higher when participants used their own mobile phones, and reported energy was significantly higher. Otherwise, people were carrying and using two mobile devices, which may have negatively influenced recording. Ideally, we want to have an app that works on every mobile phone. That is sometimes not possible due to rapid turnover of mobile apps. Thus, affirmation of this discovery of burden or interference would be useful. Studies addressing dietary intakes of pregnant women appear to be underused. Self-administered web-based questionnaires appear to be more widely used, and yet ideally these would be great to complement an image-based method, either passive or active. Now I'm going to segue to 2 to 11-year-olds. Although small samples, this is a bit of a classic. The first pass was a validation study using a metabolic research unit. The estimated weights using images matched well with actual weights. Further work done with Head Start and the home environment, not as precise, however, still came within acceptable ranges. Do note staff weighed and estimated everything. No child or family member did any image capture of foods. 5 years later, the remote food photography method enlisted parents, provided training, and enumerated intakes of 3 to 5-year-olds. And this one also, parents were trained in person, but the children actually weren't. So that leads me to thinking about, can we enlist children in helping us? And so looking at this image, how old do you think this participant is? 3 years old. So this image comes from a study in Guam among children 3 to 10 years old. The children were all loaned mobile phones to use. So looking at this image, what is your... The children were shown what to do and then asked to demonstrate what to do, given a wristband reminder, a couple of fiducial markers, and asked to take before and after images. So here's another one. How old is this participant? 5 years old. The first sample used the mobile phones during the day, so that was sample 1, while at a summer camp. Then the next step was to capture community dwelling images for 2 days. And with that sample, 2 days at 2 different times. And here's another example. How old is this participant? 9 years old. So can children take a usable image? That is including all food and the fiducial marker and images before and after. Yes, nearly 3/4 of the children were able to include both. Almost all were skilled with capturing all the food. The fiducial marker is typically captured along the edges of the field of view, lending some children not being able to capture the entire fiducial marker, but at least part of it. Children were asked if the mobile food record was easy to use, and 89% responded with a yes. Many children didn't need instruction on how to access the app. 87% of the children were willing to borrow the device again to take images of their food. Lastly, children were asked if the fiducial marker would be easy to carry around with them, and 94% said it would be easy. These are the results from the 4 days, and it appears the younger children need more reminders than the older children. Some children didn't gather any images, and this confirmed the idea of enlisting the parents, especially for the younger children. All the children received and returned the device undamaged. Children were responsible for storing the devices and the fiducial markers, and we observed the children place it in their lunch boxes, their backpacks, and pockets. Finding the best fit for children by age group is an important question to consider.
Carol, I'm so sorry to interrupt, but we do need to move to the next presentation.
Oh, okay, super. Let me do my stop share.
Okay, and our next speaker is Katie Lowe, University of Minnesota. That looks great.
Okay. Hello, today I'm going to be talking about the potential for ecological momentary assessment in dietary intake research. To start, what is ecological momentary assessment, or EMA, as I'll be referring to it throughout this presentation? EMA is a tool that was designed to collect real-time data on dynamic behaviors, psychological states, or environmental contexts. And EMA was really designed in response to a number of limitations of more traditional assessment methods. So I'm going to break it down for you and talk about each of the words and what they mean and sort of how they describe this method. Ecological. Subjects are studied in their natural environment, wherever they are, not requiring them to come into a lab or to be on a telephone. Momentary. Data is collected in near immediate state, meaning right when we want the information, asking people to describe right what they're doing at that moment. And that helps to limit or avoid retrospective recall bias. And finally, assessment. Participants are asked to complete multiple assessments per day, allowing for an understanding of within and between person variation in the behavior of interest. So how is data collected? Usually there's 2 types of assessments that are included in EMA. The first is researcher initiated, or sometimes these are called signal prompts. So what these are is researchers initiate random assessments and ask people to complete them multiple times throughout the day. And the number of times can vary based on the protocol. Usually researcher-initiated prompts assess predictors of interest, so things like mood, stress level, or context. And the second type of prompt is participant-initiated, or sometimes these are called event prompts. And these prompts, participants have to initiate. So it's when we ask participants to report on a particular event or behavior that we as researchers might not know when it was going to happen. And these are usually asked to used to assess outcomes of interest. So this is things like dietary intake, feeding practices, purging behaviors, these sorts of things. So when they happen, participants are asked to report those. So why might someone choose EMA as a method? This method allows people to study behavior in the natural environment. It avoids, or at least reduces, problems with retrospective recall bias by asking people almost right away what they were doing, what they were eating, about their behaviors, rather than asking them to recall after a period of time. EMA also allows you to study temporal patterns that are of interest to you. So thinking about those signal prompts as predictors and those event prompts as outcomes, it allows you to really test and understand momentary theoretical models that you might not otherwise be able to assess if you had longer periods of time between assessments, or if you were assessing both those predictors and those outcomes at the same time, which is how a lot of traditional methods have done it. So now I'm going to offer a little bit of an example of one study that I've been involved in that has used EMA to assess dietary intake in children. This study is called the Family Matters study, led by Dr. Jerica Burge at the University of Minnesota. I was a co-investigator on this study and helped to design and lead the EMA protocol. So I'll be talking specifically about that part of this study. So broadly, Family Matters is a study that sought to understand how family factors act as risk or protective factors for predicting childhood obesity. The study was mixed methods and longitudinal and included children aged 5 to 7 and their families. The EMA protocol was designed to include 2 different types of prompts that I'll talk about today. So the researcher-initiated prompts ask folks to report, or parents specifically, to report on their mood and stress at various time points throughout the day. And the participant-initiated prompts, this is when we ask parents to report on their own We asked them to report any time they had a shared eating occasion with their child. So it could be a snack, could be a meal, but anytime they were eating together with their child. And when they initiated one of those, we asked them questions about their dietary intake, feeding practices, and the context of the meal. So now I'm going to just show you a few examples of screenshots so you can kind of get a sense of what this would look like. So these are examples of the researcher-initiated prompts. So this is the random prompts that went out to people multiple times a day. We asked parents to report, and these are the actual screenshots that would show up on folks' cell phones. So you can see we asked about reporting of sadness or depression. We asked parents to report on various types of conflicts or stressors that might have emerged during the day. And we also asked them to report on things like their modeling. So since you woke up this morning, has Kelvin seen you check any of these things that apply? So these are things parents might be modeling. And you can kind of picture how each time someone reports on these at various time points throughout the day, they might be reporting them different, which is what we're able to capture by doing this multiple times a day. The second type of prompt was the participant initiated. So these were when we asked people to report on the meal that they were sharing with their child. These are just a couple of examples. This doesn't encompass everything we asked, but we asked about what type of eating occasion it was. We asked about context, and specifically the screenshot I offer here focuses in on the questions we asked about what types of screens or technology might have been present at the meal. That's something we were interested in. And then most applicable to this webinar today is these final screenshots, which is how we asked about dietary intakes. So we asked parents, were any of the following foods served at this eating occasion? And they would kind of tick down whichever foods may or may not have been served from this list of items. And then the following question, of those that they had indicated were served, we asked them to indicate how, which of those the children actually ate. So how well does EMA work? I have at the top of the slide here a citation of a paper that I wrote from the Family Matters data, where we were able to compare directly parent report from EMA to parent report from 24-hour dietary recalls that had an interviewer, conducting them at the end of the day. So two different methods we conducted on the same day allowed us to compare the concordance. Overall, we found concordance between the two methods to be high, but note that it did differ widely by food, with sweets having highest concordance and refined grains having lowest. In general, participants were more likely to report intake of most foods when they were reporting via the 24-hour dietary recall as compared to the EMA. One thought is that the verbal prompts from the interviewer might just help people to remember more of the things they ate. There were some exceptions to this though. And finally, concordance was highest at breakfast and snacks. Our thought is that it might be higher just when people are eating fewer foods, so it's easier to remember what they ate. And also concordance was highest when foods were prepared and eaten at home as compared to being eaten out of the home or purchased at a fast food restaurant. So in review, EMA has a number of pros and cons that should be taken into account when you're thinking if this might be an appropriate tool for you. Pros include low participant burden, and being less expensive overall when you're thinking about comparing it to a 24-hour dietary recall. Momentary data collection that's time-stamped and context-specific offers a lot of pros from my perspective. This type of data collection enables temporal sequencing of predictors and outcomes, which can help you to answer very interesting questions. And you can include questions on specific foods of interest, or for example, dietary supplements, if that was something that you were interested in understanding more about. Cons include limited details on food prep and portion size, which you saw in that very limited question that we had. And also that leaves you kind of unable to calculate the nutrient intake specifically if that was of interest to you. I think question marks for me include how much does it reduce recall bias? It feels like it should, but we haven't really directly tested that. And same with social desirability bias. So take home thoughts. Your research question matters most. You really need to ask yourself, what are you trying to measure? And do the strengths of EMA bolster your ability to answer that question? And do the weaknesses that are a part of EMA take away from your ability to answer that question? And I think I'll kind of leave you with this closing thought that I do see potential for EMA to be paired with other methods to help kind of amplify the pros and reduce some of the cons, with ideas being pairing it with digital image capture, which would allow you to have a better understanding of those portion sizes and nutrient intake, or pairing it with the standard 24-hour interview-led recalls, which might allow you to have that timestamp and some of that context, but also enable you to get some more of those details at the end of the day. And here's some readings that could guide you if you had more interest in understanding EMA. Thank you.
Thank you so much, Katie.
Yeah.
At this point, I would like to open the session for Q&A from the committee members and the sponsors. This will be an 8-minute Q&A session. And I would like to have people raise their hand. I see Anna Maria's hand up.
Hi, thank you, Amy. Thank you for those wonderful presentations. They're really quite provoking. I have two questions. One, I would be interested hearing thoughts from all three of the speakers. Carmen specifically talked about motivation, and people's motivation will differ and will affect participation. And so we've got two different age groups, I would say. You have pregnant women who we tend to think are more motivated. It's a teachable moment. And so maybe they're more motivated to actually do. And how can we use that to our advantage? And the other thing is children, right? So especially the young children. Carol, I'm fascinated by the fact that you got some young kids actually able to do this. And so once again, how can we make this a game with some kind of a prize competition that they will then feel motivated to do it? I'm not sure that we could get them to do it 7 days in a row, but I think we could come up with something. So I wondered if each of the speakers could address that, and then I'll raise my hand up and ask the second question.
Well, I know that there are some initiatives when collecting with web-based 24-hour recalls or reporting, that some kind of gaming has been introduced in data collection. So just to attract the children while they're reporting. Personally, we have conducted some research and some data collection in a small scale in school settings, or trying to engage children in reporting and like in somewhat a competition, and then adding some food preparation competition in addition to that. But that's just in a small scale data collection studies, not large scale.
All right, Dr. DeBelia, you have a question?
Yeah, thank you. Wonderful presentations. I think my question is for Katie. And I'd like to ask Katie to comment a little bit on how burdensome the EMA is for either the investigator or the family. What technology might be needed to implement that in a study? That's the first part of the question. The second part of the question is, what do we gain? You talked about it, but make it more explicit. What do we gain by adding EMA to, say, a 24-hour recall in a study?
Yeah. So speaking to the burden, I've conducted these types of studies with a variety of ages of kids, but I realized I should comment that I've always had the parents complete the EMA. So that's kind of to the previous question, that it's always been the parents completing the EMA on behalf of the child. With regard to burden, we have found that even in low income and diverse families, that this is something that they're willing to do. One of the things that we have implemented some innovations in response to their comments to make it easier. So for an example being, parents reported to us that sometimes they would get a prompt, please fill out this survey, but they would be busy right in that moment. And so one of the things we implemented was like a snooze button feature where they could kind of click a button and then a few minutes later they would be reminded, because a lot of them said, it's not that I don't want to do it, but like I'm changing a diaper, I can't do it right now. But I will forget if you don't remind me in a couple of minutes. And so we found a lot better gains when we implemented that option and just kind of reminders for them to complete them. We have asked families about the level of burden, and for the most part, they indicated that the burden to them was low. They had their phones with them. They're used to engaging with their phones throughout the day. And so it wasn't something that they felt, they wanted it to be on their phone. They didn't want to have to carry another device. That was another type of burden that they felt like would be too much for them. So I feel like
Oh, I was just going to ask, how long did they spend interacting with it? How long does it
Yeah,
take?
yes, I saw that question in the chat. So for the most part, we kind of best practices is you try to keep each assessment 2 minutes, 2 to 3 minutes or under. So it's very quick. So you really have to be, I think the burden is on you to be very specific about what you want to know, and then to design the questions in such a way, and you can look back at the screenshots, but we design the questions in such a way that they're really just like ticking down boxes or sliding a bar or radio buttons. It's never something that is more difficult to engage in than that. So making it, and it's got to fit on their screen. There's a lot of that kind of stuff to reduce the burden. And kind of that, I think, speaks to the technology question that you had about how you incorporate this. We, in my particular department at the University of Minnesota, because we do a lot of this EMA research, we have a computer programmer who helps to create these things for us. But I have also worked on studies where we engage an app. So there are apps that you can, that you have to purchase either the app or you pay a fee to have the researchers who run the app to help you to set up the EMA. So I certainly feel like if I wasn't in my department, I would be using an app and not doing it myself. I happen to be lucky enough to be in a place that's supporting this type of research with the computer programmer. And then the final question, I guess you asked about how it might add to recalls. I think the recall, at least at this point, is always going to get you more details about what, like you can dig in and you can ask about portion size and food prep and type of milk and, all these things that you can't do on EMA. But I think the context is so interesting. I think people eat differently when they're different places. I think mood can really drive and impact how somebody eats or how somebody engages with food. And by being able to really like timestamp that information, I think it just allows you to answer different types of questions.
So it helps you answer a different question, or does it help you with the biases introduced by the 24-hour recall? That was the
Yeah, that's a great question. I mean, I have a hunch that if you were able to, I have a hunch that people sometimes forget that they eat food. And so something I'm interested in exploring is if you were to have somebody even click a button or just indicate, that participant prompt, I ate something, or maybe tick down the boxes of kind of what it was, that then if you paired that with a 24-hour recall at the end of the day where you could dig in and say, tell me more about that food, how much of it that you ate, that it might kind of marry those two methods of like, it would help reduce the retrospective recall bias. But if you really want the details, you need the 24-hour dietary recall to do that. And so my sense is like the combination of those things could improve our assessment of the intake.
Thank you so much.
Especially of those and I think that it's a different type of food that you forget eating as well, right? You remember the meals you eat, but you forget the stuff you like grab when you're walking by your coworker's desk, or you eat in your car, the goldfish crackers you hand your kid when you're driving someplace. So I think it helps you to capture those foods a little bit better.
That's great. And I look forward to hearing more about statistical methods in this next session. So we'll move now to Dr. Diane Catelier of RTI, who will talk about harmonization of methods for diet assessment.
Okay, thank you, Alice, for sharing my slides. I'd like to start with thanking the organizers of this webinar series for inviting me to speak about harmonization of dietary constructs. So next slide. So inherent behind the need for data harmonization is this recognition that data sharing and pooling of data across studies allows scientists to expedite the translation of research results into knowledge to improve the human health. So even the International Committee of Medical Journal Editors went further to suggest that given the significant human and monetary investments made for research, it's actually an ethical obligation to share data, provided it can be done responsibly. So many government entities, including NIH, are investing heavily in activities to facilitate data aggregation and harmonization, and in tools and platforms to make that data accessible to the wider scientific community, so that the data collected through public funds can be used to address innovative and high-impact research questions that require a lot of data. So NIH has echoed calls to put harmonization into action in the area of child health through the Environmental Influences on Child Health Outcomes Consortium. ECHO is a consortium of 31 ECHO awards comprising 69 racial and ethnic and geographically diverse cohorts that contribute data on approximately 35,000 pregnancies and 58,000 children. The primary objective of ECHO is to facilitate the creation of an ECHO-wide cohort data platform that consists of data from all cohorts to help understand the effects of early life environmental exposures, primarily from pregnancy to age 5, on child health and development. Next slide. So this leftmost side of the schematic shows the exposures of interest that span the physical, chemical, social, behavioral, and biological domains that include diet during the prenatal life stage and throughout the life course from early childhood through adolescence. The outcomes span pre-peri and postnatal outcomes such as birth defects, prematurity, and mortality, and neurodevelopment, airways conditions, obesity and obesity-related conditions, and an outcome related that we refer to as positive health. So next slide. When considering combining data across studies, even when the same measure is used, differences are inevitably introduced related to the population, the language, the method of administration, and processing algorithms. So first, it's important to take stock of all variation in processes and procedures related to the data collection of the measure you intend to harmonize. Next, you need to specify metadata that will allow the end user to select eligible studies based on the specific research question and evaluate the effects of measurement methods and data collection procedures on the overall results. So the metadata step is critical. The final step is to evaluate the success of your harmonization using analytical methods to explore heterogeneity of results and imbalances in the sample size due to over-influence of a particular study or subgroup. So a couple of caveats: harmonization is not for the faint of heart, can be very time-consuming and labor-intensive, and it's difficult to undertake if there are inadequate resources for the participating studies, both in terms of data management and investigator time. So ideally, a team approach should be undertaken with involvement from statisticians, the investigators involved in the original data collection, and subject matter experts. Next slide. So understanding your research question is integral to harmonization. Here I've listed some examples of types of dietary measures that might be considered for harmonization or dietary constructs. And in the remainder of the presentation, I'll provide examples from the ECHO experience related to harmonization of energy intake, nutrient intake during pregnancy, and diet quality in early childhood. Next slide. So I've listed some of the factors that should be considered when trying to determine the compatibility of diet data for aggregation. In terms of the population, harmonized measures are unlikely to be invariant across life stages, as aspects of the underlying diet instruments will vary across those life stages. So for example, the block FFQ is a proxy report in early childhood and a self-report in later stages, life stages. The data collection protocol is critical. The number of times, for instance, that the 24-hour recall is administered, or for the FFQ, the time reference for recall. Is it the past year, the past 3 months, or since the woman has become pregnant? The mode of administration, is this online, by phone, or in person? And the actual instrumentation is probably the most critical. Is this a food frequency questionnaire, 24-hour recall, remote photography? Is it a dietary screener, or is it the full administration of the recall method? And again, this is subtle, but changing instrument versions and the nutrient databases that are used for analysis do change over time and should be taken into account. So next slide. So the diet data available in ECHO very quickly are 24-hour recalls, both from the ASA 24 that spanned 4 different versions, and the NDSR, food frequency questionnaires. There are a couple for just during pregnancy, the Harvard FFQ and the Block 2005 FFQ. And even there, the reference period, like I said, was differed almost for every cohort that administered the questionnaire. There were some enhancements over time that included inclusion of vitamin supplements, some additional questions that related to omega-3 and the microbiome. We also had Block FFQ for the child diet, full versions for the 2 to 7-year-olds and 8 to 18-year-olds, but there was also a screener for the 2 to 18-year-old time period. And finally, we have the NCI diet history questionnaires. Again, wouldn't it be nice to have 1 version, but no, we have 2 versions. So this was a challenge, or has been a challenge, continues to be a challenge. So next slide. So the first example I'd like to use is one in which we're trying to harmonize methods to determine diet quality. And here, where the diet quality measure we were primarily interested in, at least to start, was the Healthy Eating Index 2015 version. So the steps involved doing a crosswalk of output from 24-hour recalls and platforms. So the ASA 24 across the different 8 versions, as well as the NDSR data. And then a crosswalk of output from FFQ versions and instruments, the block, the Harvard FFQ, the Diet History Questionnaire. The Diet History Questionnaire. But just to note, and I've given the reference here at the bottom of the page, there are some algorithms that are provided, example algorithms that are provided on a website for how to do the HEI calculation if you have recall data versus FFQ data. I will note that in this first pass anyway, it's hard to do otherwise, we had to go with the nutrient database that was for which the data were provided. And again, at least to start, most of our data is extant data, not new data. And we did not use dietary screeners. There's just insufficient information to assess diet quality with those. Next slide. So here we show the distribution of 2 measures of the 13 HEI scores. On the left-hand side for children ages 2 to 7, and on the right-hand side, women during pregnancy, that were derived from different versions of the ASA 24 and the block FFQ, just for simplicity. While we show the p-values for the tests of equality, they're all statistically significant with the exception of the whole grains for children 2 to 7. In general, I think it's better to do a global test for comparability across all 13 HEI scores, as it does control for your type 1 error. So in ECHO, we're at the stage of primarily dealing with extant data. So there could be an opportunity to have cohorts collect data both on the block FFQ and the ASA as the presumed gold standard in a sample of participants that would allow us to use regression calibration methods to correct for some of the measurement error we're showing here. Next slide. Now switching gears to research around diet quality, I wanted to present some work from Kate Souter and a team of co-investigators that explored sociodemographic and obesity-related disparities in the risk of inadequate and excessive intake of micronutrients during pregnancy. We had 15 prenatal cohorts contributing data on about 10,000 pregnant women. 5 of those cohorts provided 24-hour recall data and 10 provided FFQ data. So individual cohorts calculated average daily intake using appropriate nutrient database and dietary supplement labels. We did not combine data across methods. We assessed the differences in mean intake for nutrients between methodologies, recall versus FFQ, and all the tests were statistically significant. Next slide. Despite the use of different methodologies and nutrient databases across cohorts and over time, the analysis that was based on food-based results were similar between the methodologies in terms of plus or minus 10% for most nutrients, including the directionality of the disparity analyses. Results for dietary supplements varied more between methodologies, but that was a function, I think, of the subset that actually had data on supplements. So next slide. And this just shows really the data behind those conclusions. So I'm going to skip the next slide, and seeing that we're at time here, and just go to some final thoughts. It's extremely important to document your harmonization methods for transparency, apply tested heterogeneity to identify factors that have an effect on the diet outcome of interest, stratify if appropriate. And you really need to think about the trade-offs between limiting your analysis to a single measurement versus harmonizing that relate to generalizability or selection bias, measurement error, and power. So certainly apply sensitivity analysis, and one simple way to do that is a leave one out analysis, either by measurement method or by study, and that's beneficial to understand the potential impact of the overall findings. And as always, you must account for the between study heterogeneity. Apologies for going over, but I would like to turn things over to Angela Lees from the University of South Carolina.
Thank you so much. Can we have the slides? Yes, they're coming. Wonderful. Well, thank you. This was the perfect talk to precede mine. And I'll be talking about matching the research question to the appropriate diet assessment method and statistical analysis. And I really want to focus on two primary themes, which is one, the concept of multidimensionality, considering food groups and nutrients jointly in epidemiologic analyses. And the second theme is integrating measurement error into the design and analysis. And can we have the next slide? Wonderful. And can go on. Okay, actually, let's go back one, sorry. So just a word about defining multidimensionality, as that might not be that common. So when we nutritional epidemiologists talk about multidimensionality, it is really with the concept of having many foods and nutrients and other constituents be represented in the diet that we're trying to study. And each of these, the food, a nutrient, or other constituent, could be considered a dimension onto itself. Next slide, please. So the three research questions that I want to address in these next few slides that focus on the attribute of multidimensionality are the following. How do different nutrients or foods potentially interact in their impact on health outcomes? And what does the food source of a nutrient tell us about maybe this nutrient's effect on health? And then lastly, what can the diet quality indices tell us about multidimensionality? Next slide, please. So I want to start out with a very simplistic approach to study multidimensionality that's been around a long time, but that's still, I think, underutilized, which is the traditional interaction model. This is a study by Susan Steck, a colleague of mine, that has focused on predictors, dietary predictors of postmenopausal breast cancer, and with a particular focus on the intake of grilled, barbecued, and smoked meats, but stratified by fruit and vegetable intake in the last year. And so they in this study actually formally tested the interaction between fruit intake and this particular type of meat consumption, and found that only in the women who reported low fruit and vegetable intake were these grilled, barbecued, and smoked meats actually associated with an increased odds of breast cancer, whereas those in the women that had very high or regular fruit and vegetable intake, there was no association. So this is a typical example of what we would call effect modification or interaction. Next slide, please. A different approach to studying this concept of multidimensionality is this food group-based approach to studying the impact of polyunsaturated fatty acids and their associations on LDL cholesterol in youth with type 1 diabetes. This is from the Search Nutrition study, and which is part of the Search studies, and it is focused on youth age 10 and older. And in this analysis, what we did is we basically backed into the underlying architecture of the FFQ and derived the specific amount of Pufas that was delivered by different food groups. And the 5 food groups that contribute most to Pufa intake in this population are listed here in this table, and there are many more that we included in our modeling. But what I just want to point out is that not all Pufa delivering food groups actually were associated in the same way with LDL cholesterol. If you see in this slide, it was only the Pufas from nuts that actually had an inverse association with LDL cholesterol. So here, the Pufas were a really good thing. And the Pufas from refined grains were actually also significantly associated, but in a positive direction, as were the Pufas from high-fat chicken, which is largely due to chicken nugget consumption. So if you can click to the next slide, what we then did is we ran a substitution model, and the next slide. And what you can get from this kind of approach is a sense of what would happen if a person replaced their Pufa intake from high-fat chicken, say, with Pufas from nuts. And in this study, that correlated with a reduction of 7 milligrams per deciliter in LDL cholesterol, which is quite a significant reduction. So this is this food group-based approach, which I think could also be used more in studies with a focus on nutrients. Next slide, please. And so now I want to turn to the diet quality indices and share a little bit of data from some ongoing work of this group. We've been fortunate to work with colleagues at the National Cancer Institute for many years on dietary patterns. And this is a paper that actually very nicely summarizes some of the frontiers in dietary pattern research. And if we go to the next slide, as Diane has already mentioned, I mean, there are quite a number of diet quality indices. The H Healthy Eating Index, what we're looking at here is a depiction of the Mediterranean Diet Index. And it's a radar graph where if you were to eat a perfect diet following this index, you know, your intake would be around the perimeter, right? So the scale is 0 to 100%, and you're seeing the visualization of intake levels. You would be seeing them at each level. What these group of authors led by Buckland found was that when they actually tried to study the individual components of this index to figure out which of them were predictive of mortality, that only olive oil and alcohol ended up being predictive. And the question that raised is, what really has to happen if you're already studying a concept of total diet quality? What has to happen if you want to increase somebody's, say, olive oil intake or alcohol intake while holding calories constant, while holding all the other components of the diet quality predictors here constant? The reality is you would have to find foods or nutrients that are not considered in this indicator at all, and that are and you're still controlling for calories, right? So in this case, it would basically come down to something like changing potato intake or changing added sugar intake, because those are not part of this Mediterranean Diet Index. Next slide, please. From our own work, just a couple of slides trying to identify some of the commonalities and differences in meeting these HEI 2015 recommendations, still focusing on the multidimensionality question. And you can see this kind of a spaghetti-like radar graph. And the question is, are there some patterns, are there some commonalities that we could identify underlying this? And the fact is, yes. We used a technique from medical imaging processing called shape analysis to identify so-called constellations underlying these data. And you can see that there are some distinct differences between the two constellations on the left and the two constellations on the right. So this is another way to think about studying multidimensionality. Next slide, please. So now shifting gears very quickly to the topic of measurement error correction. Next slide. We've already heard a lot about how measurement error, or simply measurement noise, affects nutritional epidemiology. And I think that's expressed expertly in this quote from Larry Friedman. Next slide, please. We had a diet assessment study funded within SEARCH and the SEARCH Nutrition Ancillary Study, where everyone, in our main study completed a food frequency questionnaire. This was the modified block questionnaire with some race, ethnic, and geographical cultural informed foods added in our population of 10 and age 10 and older kids. So in addition to the main instrument, we had in this diet assessment sub-study another FFQ and 3 24-hour recalls in a small study of less than 200 kids. And we applied some of the measurement error correction methodology that's been developed by a variety of statisticians, including those at the National Cancer Institute, to this question of how are sugar-sweetened beverages associated with lipid levels in our sample of youth with diabetes. Next slide. And there's a whole seminar talk on this NCI method that I just wanted to bring your attention to. Next slide. So in our sample, we found that this measurement error correction actually made a huge difference in terms of the outcome. if you focus on the left block of data, what's labeled the naive model estimates, no measurement error adjustment, you can see that the difference in a very low intake of sugar sweetened beverages to a moderate intake of half a serving a day is only about 1.3 to 1.4 milligrams of these lipids, triglycerides, and cholesterols, which is not a clinically meaningful amount, right? After measurement error adjustment, these differences actually increased to 3.7 to 4 milligrams per deciliter, which is really a very nice effect. So I think this is another reason why I'm very excited about these methods of regression calibration, because I think if we have the opportunity to apply them more, we will also find stronger associations in our research. Next slide. So in conclusions, I think this joint consideration of multiple nutrients or food groups and combinations thereof is really underutilized in nutritional epidemiology. And the increased focus on multidimensionality is a really promising area of research. And then lastly, as I just said, the widespread integration of measurement error correction methods into the design and the analysis of dietary assessment studies is, I think, something that is really needed to advance the field. Thank you so much.
Thank you very much. We really appreciate these two presentations. And now we will open things up for a short Q&A from committee members and sponsors for the prior two speakers. So if you're a committee member or sponsor and have a question, I see Dr. DeValia again, so I'll start with you.
Thank you very much. Wonderful talks. Angela, a question for you. Can you give us an example of research questions? Your talk was about matching the research question to data analysis and data collection. Can you give us an example of a research question you need for a further conceptual development of how to use the multi-dimensionality framework to address that question?
Yeah, I think there are lots of questions like that. And one of them that I think about a lot is a question that comes up when we try to translate dietary intake guidelines, right? So using the HEI as an example, you know, we could very easily say from a population perspective, the recommendation needs to be everybody eat this, you know, really good diet, largely plant-based. And we can do the research to figure out what the effect of that on a health outcome would be. But the multidimensionality comes in when then, you know, a typical consumer question bubbles up and somebody asks, I really like my hamburgers, right? I really want to continue eating hamburgers because they make me happy. And what is the impact of continuing my red meat consumption when I'm going to start adhering to the rest of this healthy eating construct? And that actually, that puts you right in the realm of multidimensionality.
Thank you. Anna Maria?
Yes, hi. I've got a question for Diane. Good to see you again, Diane. You know, I find it very interesting that in the US, and I'm not sure about Canada, but, you know, we see a lot in European countries where people are coming together and creating more consortiums for being able to answer questions across different databases. But we don't seem to do that as much. ECHO is one example where we've gotten that, but even ECHO's example, there's lots of people who've got dietary data. How can we get to a more open source in being able to share these data? And how could we then be able to provide more protocols for the harmonization of these data to see whether or not, in fact, our picture of, you know, what we think pregnant women eat in the United
I think you got cut off at the end there, but I think I caught your gist. I do think that there have been other consortium that have come around together around the desire to standardize data collection methods and come up with kind of a common data model of platforms for being able to best combine and most quickly be able to put data together on a platform that is usable by the scientific community. I don't know that that's possible until there is a group that is speaking from one voice. I think ECHO is one challenge, and it's been really, really challenging getting all the data on the platform. It would really benefit from having a group that could prioritize what would be the way to collect data, best collect the data. And I think there are biggest challenges in terms of supplement use that could really use some guidance.
So I'm sorry, I think the last part of my question was the fact that are there protocols that could be established and shared widely for the data harmonization? So in order to make these efforts not be so heroic, I guess.
Yes. Yeah. Well, one of the things that we're making available as part of ECHO is kind of this crosswalk that does take all of the source data and says, if you're using any of these measures, this is how it crosswalks into the inputs that go into developing not only the healthy eating index, but other dietary quality measures. And so we would like to make that available and hopefully streamline the process.
Diane, can you say something about data repositories for these types of data?
I'm not aware of larger data repositories for diet data. These data are expected to be made public in 2022. Generally public, it's right now being limited. This is one of the challenges, I think, to data sharing, is that there's so much investment made from the investigator in collecting these data that it does warrant having a little bit of time for them to do their own analyses before making them generally available. But
do we have other questions from the committee or sponsors before we go into the final panel discussion? I see one in the chat from Dr. Dwyer asking about, actually a couple. Maybe Diane wants to talk about one, and then we can have Angela weigh in on the other. The first, just about how challenging it is to harmonize supplements across the FFQ. And Diane, I see you've started typing an answer, but maybe you want to address that.
Yeah, I couldn't agree more, Joanna. Harmonization of the intake from supplements is really challenging. There's variation in the supplements that are included, and not only that, but the level of detail about the nutrient composition in the supplements can vary across the measurement methods. I think this is an area where standardization is needed across the FFQs and could use some general guidance.
And then she had one more question about use of multidimensionality in the context of multivitamin and mineral supplements and other multi-ingredient dietary supplements. Angela, did you have anything to add?
Yeah, full disclosure, I'm probably not the best person to answer that question because I've not worked extensively in the supplement domain. But I will say I do have an appreciation for how complex it is to even quantify the supplements properly. And given that they're inherently, I mean, most supplements include multiple agents or constituents, I would think this is really a very tough issue when you're trying to do something like the Pufa analysis we did, right? Assuming people wanted to consume Pufas from supplements, I just imagine the overlay of that on top of the food group based approach. I think it should be doable because at the end of the day, it's just sort of a layering that I would conceptualize the supplements as like another layer of information. But I will say I have not spent any time thinking through exactly how to implement that. So.
Well, thank you so much for such an engaging discussion. And now we will move to our special panel discussion. This discussion will feature some of the speakers from previous sessions as well as in the current session. And I'd like to welcome Dr. Roberto DeVito of Brown University to our panel. And in addition, from prior sessions, Drs. Anne-Sophie Morissette from Laval University, Daniela Sotras from University of North Carolina, and Tom Baranowski from Baylor College of Medicine are back to join in this panel. So we'll take the first bit of time for questions from the sponsors and from the committee members, and then we will open it up to general questions in the Q&A. So I'll ask all the panelists to come back on. And then I'd just like to kick things off talking about, you know, we've seen some really cool and innovative assessment methods, and asking the full panel, which methods do you think have the most promise for assessing intake in pregnancy in ages 2 to 11? And what kind of research and evaluation is needed before these methods are ready for prime time? And what are the barriers to making this type of research happen?
So I'll take a first shot. It seems to me that we can't predict what the methods are, that technology is advancing at such a rapid rate that who knows what's going to be useful in a couple of years. Carol has a preference currently for the active approach to objective assessment. I prefer the more passive approach but there's huge variety currently, and even within our variety, there's rapid advances being made. So I don't think that that we can assess. There's a 5-year horizon and there's a 25-year horizon. And I think the 25-year horizon is completely wide open about what we're going to use. I think that we need to. It's been unacceptable now for a couple of years in physical activity assessment to use self-report methods because of the huge inaccuracy. And that's been instigated by the use of accelerometry. Currently, as in dietary assessment, more and more technology methods are being used. My guess is that it's going to be unacceptable to take self-report methods in diet. But you know, that's a 25-year horizon, not a 5-year horizon.
Anna Maria?
I wanted to follow up with that question. It's a great sort of segue to what I wanted to ask Carol previously. In the sense that, you know, we are nutritionists, we, you know, try to get after accuracy in assessing diet. And yet the technology, and especially when you think about the crowdsourcing technology on the use of apps these days. So, you know, you Google on your phone and there are a ton of apps for pregnancy, and I'm going to focus in on pregnancy. And the majority of them include some form of dietary assessment. And so my question is to those of you who wish to answer it, at what point are we going to realize that maybe we go ahead and partner with one of those apps and decide to use that, but then as, you know, Kate actually brought to our attention, we can maybe sort of complement the information that we're getting from the apps with a 24-hour recall and combine the data to be able to enhance our assessment for research purposes. I just feel like we've been in this field for quite some time and we're spending a lot of money on trying to increase precision, and we might not get there. And there's a lot of other people outside of our field that are more savvy than us.
I think, I think what you just said is spot on. And one of them has to, one of the points that you made is that you made this idea of combining methods. And I think that that's something that we really need to be open to and think, because each of them has their own positives and negatives, but by somehow, if we can harmonize them to, you know, put them together, I think it would be, that's one of the ways that we should be thinking of going. And I think that's a really good idea. And you know, some of us do that, I think, unconsciously. And then with regard to Tom's comment, I'm open to all the methods, Tom. The only thing is that right now, some of the, you know, being able to meander through this just long stretch of information that about 90% of it you have to throw out. You know, I still haven't been able to think of doing that on a regular basis.
But there are algorithms that have been artificial intelligence algorithms that limit the number of images that you got to go through. Now there's error even in those algorithms, but I 5 years from now, a lot of those problems are going to be solved.
Well, along those lines, I was wondering if Roberta or Daniela or Diane wants to weigh in on statistical or other techniques that are commonly used in other fields that might be informative here. So we heard in a previous webinar Nick Horton talking about multiple informants. Are there other approaches or methods that we should be thinking about?
Yeah, I think that, I mean, this comes to at the right time, in the sense that right now we are in this big data area for all the data, but of course also for nutritional epidemiological data. And I saw this amazing talk with Carol regarding this food image, and I was quite impressed because I never seen something like that. I'm used to work with nutritions and nutrients or food items, and I think that this could be a very good idea to fill sometimes the gap regarding like harmonization method. So right now there are like many different problems also to harmonize the different data from also different population or also different like informants. And I saw in nutritional data people use and try to or stack the data in one big data set and perform factor analysis. And this is also another different field, like in genetics, to, for example, find common characteristics across population or across different strategies. Or another way to do that is to perform separate factor analysis in each of these data or in each of these methods. To capture the differences. Like recent, also I developed this multi-study factor analysis, and we are work also with Daniela with this in nutritional epidemiological data, where we can capture commonalities across different and multiple platform or multiple data, but also study specific. And I think that this could be a good direction to go toward. In the sense also with this multiple technology like food image and nutrients. I mean, we need some method that capture the reproducibility across these different and multiple platforms, but also the specificness, so this heterogeneity across these different and multiple studies or platform. That was.
And yes, I can add to what Roberta was saying. So yes, I'm convinced that we need to combine several methods because, as it was mentioned before, each one has its strengths and each one has its limitations. So we just need to kind of combine them all together, and then rather than Roberta was saying, or stacking or combining them, we just need to model them simultaneously, jointly, because if we just put everything together, then we end up like combining different sources of measurement error or different, rather than fitting some joint models, which brings me to the other part. These models are available and are already developed, but the problem is making them available in an easy way to other people that can use it. So that's one of the advantages of using the NCI method, because there are macros that people can use easily if you're working with a statistician. But nevertheless, that will correct some of the intra-person variability. But these, if we're just using self-reported data, it will still be biased. So we need to bring up either biomarkers or different sources of self-reported, and that we can model this simultaneously and have the capability of have these models that are available, but more friendly, so more other people can use them.
I can just interject for just 1 minute, just saying, agreeing again with both sets of comments. I think that the availability and the technical requirements for doing some of these more latent variable models or factor analyses are it's pretty challenging, but if you can make it a more user-friendly platform, that would be great. I would also hope that we could move towards a protocol where there is some multiple data collection, where for certain subset of participants, we get data on across methods, so that we start to understand how these are interrelated. It doesn't have to be a lot of data, and particularly if you're getting those kinds of repeated measurements across different studies, that would better allow us to be able to fit some of these models, more sophisticated models.
Right, I'll turn it over to Dr. DeBellia now, who's been patiently waiting.
No, this is a fascinating discussion, and I just want to ask this question, which might be, I don't know, provocative, but I'm wondering, to what extent do we have studies or groups of studies such as ECHO, maybe more than ECHO, like Anna Maria was suggesting earlier, whereby we could kind of get the data that's already out there and develop these models that the statisticians were talking about, and then fed them back to the world so that every study in the future doesn't have to repeat, the whole shenanigans of collecting diet by 2, 3, or 4 methods to correct for measurement error, but maybe they could apply, a simpler, self-reported even way to collect diet, because there will be ways to correct for measurement error and address bias. What is missing? Why can't we do that? I mean, for sure, ECHO is a large collection of already collected dietary data using multiple ways of data collection, including biomarkers and microbiome and self-report and technology-based methods. I understand it's going to take some time, but isn't that a way to address this conundrum?
I think we're moving towards that. So it's been now with technology and having these cloud-based platforms where we can have data loaded and secure enough so that people cannot download it, but they can analyze it. And so as we move forward in that direction, data will be available from different sources so that anyone, it's like right now with the All of Us program, data is available there. So obviously people need to know it and need to use it. And as more studies are putting their data available, like they were saying, some of the repositories or the effort from the Common Fund that are trying to all the data to be on the platform, that anyone, any researcher that applies to do an analysis can use this data that it's available.
So how close are we, from a surveillance perspective of being able to harmonize data across, large groups of studies, as Dana mentioned, to get a picture of the overall nutritional status of our population, or, pregnant women or children 2 to 11. I don't know if Anne-Sophie wants to weigh in on that too, or Diane. What are the barriers and the biggest challenges to being able to make a statement like that for the whole country?
I'm not sure if I'm the best person to be speaking of this. My experience is somewhat limited to ECHO, but I do think that one of the prerequisites is kind of coming up with this common data model where, like when you submit data to dbGaP, there's an expectation that it comes with a data dictionary, everything's going to be specified in a certain way. And pushing that out onto the cohorts, because certainly the repository doesn't have the resources to ensure that those data come in that way. So in that sense, we're a little bit ahead of the game, since so many of these measures, the FFQs and the 24-hour recalls, do come somewhat in a standardized format. It isn't tremendous to have data managers put it into that format, but I do think that there needs to be that specification in advance, and any kind of metadata that is required to go along with it.
Anna Maria?
I wanted to follow up with the use of being able to collect information on young kids. And Carol, I really was fascinated by those pictures of having young kids being able to do this. is this something that we should be relying more on the fact that we can engage younger children? Because the norm has been, or I should say the accepted belief was that, you always needed a parent until about age 11. That might be pushing down a little bit more to 9, but are we missing an opportunity to collect more information from younger kids? And I leave that to you and anybody else who might want to chime in.
That's a good, excellent, really nice. It's a really good point. I am amazed at how much children can do. And what they're doing, of course, is they're mimicking the people around them. So when they see people around them using, taking images, then they want to do the same. But I do, so I'm really in favor of tapping into children as much as possible. Where I draw the line is at the 2-year-old age. They still are distracted by a lot of things. So we had some problems with the 2-year, the really young ones, because they'll leave the mobile phone wherever they last were. And so you have to find it. So we have to, I hadn't thought about this till just right now. We should have put little sensors in them, that we could find. But at this point in time, when you think about it, that these children, they're getting exposed to these really young, and they can actually handle doing these activities. I was surprised by it when we first started, but now I'm convinced. However, we do need to have, the parent still needs to do some reminding, but for the most part, we should be tapping into the whole family. Make it a family affair. Be an interesting approach to do too.
So if I can jump in and sort of add something, I think that this is another opportunity where we have to not assume things can only be done one way or the other. I think for a long time it was, we can't count on kids to report. So when they're at school, we just won't know what they ate. And so it's sort of like, it may not be as accurate. We may not get all of the details, and yet we really don't have much to lose if the alternative is not knowing at all what they eat. And so thinking about, it may be we want the parents' engagement when they're at home, but when they're not at home, we rely on the kids, and we know, gosh, there might be some error, but it's better than not having that data at all.
I had a question for Carmen. She had a slide that we weren't able to get to about plate waste. And I wanted to draw you out a little bit about the quality of intake information we can get, we're currently getting for surveillance. Is that adequate to inform about plate waste if we think about a sustainability perspective?
Yeah, thank you, Amy. Yeah, plate waste is something that is useful in terms of sustainability. This is something that is coming more and more, and most of the research conducted in this area has been based in terms of greenhouse emissions or water use or whatever. But certainly there are other dimensions of food sustainability that haven't hardly been addressed. And by taking pictures before and after, this kind of analysis can be conducted. So this kind of information can be taken at any age and any population subgroup, and will inform about the kind of foods which are being wasted and so on. But in addition, as has been reported, that regarding parents and children data intake reports, mismatches can happen and has been reported that parents report on the food portion served rather than the food portion consumed. And by analyzing food served and wastes, then some kind of error analysis can be conducted. So it's really promising and useful information.
Thank you. And now I just want to remind our listeners that we are opening things up to general questions in addition to questions from sponsors and the panel. And Susan Johnson has placed one in the chat that was actually the first thing I thought when I saw Tom's webinar earlier in our series as the parent of 3 boys. And that's, if we have children collect these data, how do we assure that the images won't capture anything that shouldn't be captured? And I was thinking about this camera taking a picture, with frequency not controlled by the student, and all the fun that boys can get into. And Susan also notes boys don't have, or maybe children don't have much discretion. So what kind of safeguards do we need to have in place for use of these, especially passive technologies?
So in the studies we've done, the images are encrypted immediately upon taking. Once they're transmitted to our central location, we de-identify the images, so you can't tell who's being done, who's doing what. And everybody in the family consents to those kinds of procedures. And there's an off button so that if the child doesn't want images to be taken at a certain period of day, they can turn the camera off. So we've only done 30 kids, 2 days apiece. And while we had some concerns as well, we didn't come across anything that you wouldn't share in a family situation, what was recorded on the images.
So how do you handle, for example, a kid going to the restroom at school?
So the child can turn the image off. It's not like we share the images with everybody in the public school. It's just analyzed at that time by our dietitians, and they're adult women, and they weren't particularly offended by those kinds of events.
So you didn't get pushback from, for example, other parents in the class or anything?
No.
Thank you. I had you want to ask a question, Ana Maria? I'll let you jump in.
Well, I know we have some other questions, but I was just gonna preempt folks because we got an amazing panel, a group of panelists here. And I was wondering if you had to sort of summarize what you would say is your recommendation for how we should move forward as researchers in this field. What would you recommend right now? And we can break it out from pregnancy to children and be separate if we need to, but it would just be interesting to sort of hear everybody's thoughts on that.
So I think that we need to take a really long-term perspective, that the methods that are currently in use are flawed. Self-report error is enormous. 50% error in some of the work we've done with children. The statisticians are spending enormous amounts of time trying to correct for that errors. Wouldn't it be nice if we had virtually error-free data going into the statistical analysis that's being done? So I think that the people concerned about dietary intake assessment, mostly dietitians, need to generate interesting hypotheses about methods of data collection, create teams of investigators, including engineers, including big data people, including anybody else who needs to be involved. And let's get on with the business of improving the methods by using technology in dietary assessment. And that's a 25-year perspective, not a 5-year perspective.
Go ahead.
Oh, sorry. No, I would like to add that from a statistical perspective, also with this amount of data, it's really good to go toward the direction of building strong model that also take care about all of these technologies. Like I know that with these, for example, image, there are these fMRI data that use machine learning like sparse principal component analysis that can remove the signal that it's not I mean, the good signal, that it's like the error. So that could be also nice to go in the direction of, I think, sharing the data, integrate the data, like measure the reproducibility. So the strong signal that is reproducible across these like multiple studies and multiple like informants. I also follow the talk, the amazing talk of Nick Horton, I think it was in webinar 2, regarding these multiple informants. And I think that one major concern was also the missing data problem. Having these multiple integration, multiple studies can also help us to assess without error or with less error this missing data value.
So I wondered if we could get some people to talk a little bit about, in previous webinars, we've pondered biospecimens. So either biomarkers or samples even of the microbiome. Are these things that could be used in order to help us correct for any error and biases, especially in younger children who might not want to give blood samples?
I'll just say that I think, yes, we need to move forward and try. Obviously, right now, there's just a handful of biomarkers that are available. So until we get other biomarkers for other nutrients and foods, we're still far away from that. So one of my concerns about the pictures, especially the passive, I like the idea, but the active, I wonder whether the child will take the picture, will spill out everything first, and then take the picture. The after, I'm still concerned. Yes, it's a picture, we'll be able to see it, but it's still self-reported because we don't know if it gave it to the friend or just toss everything to the garbage. So I think it's good, but still far away from it's just self-reported.
I wonder if Angela, have you sort of ventured out into using sort of the microbiome to sort of be like having the capability, being able to triangulate data if you've got self-report, biomarker, microbiome?
We have not, and I will say why not. I mean, the microbiome is not an area that I've ventured into at all, but we've been thinking about integrating some biomarker data with just self-reported dietary assessment. And I think one of the really difficult issues is that, you know, with each step inward into the human body, there are more factors that determine the levels of whatever, say, nutrient you're studying, that have their own structures associated with them, their own metabolic controls. And I think you have to be very thoughtful about assuming that a nutrient measured inside the body is telling you everything you want to know. I think it doesn't. I mean, I think it actually means you're studying something different than when you're relying on self-report. I'm not suggesting that it's not doable. I'm just suggesting it has to be very thoughtfully constructed, because we are getting into the pathway of what happens with the food after ingestion, which means there are all of these other systems invoked that will have their own error structures, et cetera, et cetera.
Diana?
Me?
Yeah, oh, sorry.
Sorry. So I earlier, a few minutes ago, I thought I heard some potentially different directions suggested by the members of our panels. Tom was talking about, forget about using biostatisticians and analysts to correct for measurement error after the fact, invest into making these data collection methods more reliable and more accurate, but it's going to take us 25 years to get there. And then I heard Roberta and earlier Daniela, I thought, kind of making the case that we are close to developing, based on data already collected, strong models and calibration methods that we can address the issues post data collection right now or soon, based on where we are with the development of these models. Am I understanding these two potential divergent directions clearly? Can you comment on whether they are divergent or not, and also the timelines for which one we can get at faster, sooner?
Can I comment on that? So I think if one takes a step backward, in other fields of science, it's very commonly done that one uses multiple different informants or measurements and integrates them. Because I think from a statistical perspective, everybody understands that if we have more data, an average will be closer to the truth, right? But I think we've been very resistant in many subspecialties of integrating data from different sources, because we always think there is something like the truth. I think the longer I've been in this field, the more I believe there is no such thing really as the truth or the perfect instrument. But I think an approximation of using very good instruments combined will get us so much closer than just saying, well, I think the FFQ is the best instrument. I'm just going to stick with it, right? That's no longer not that it ever was my position, but it certainly wouldn't be nowadays. So I think more openness to the integration of data at the collection stage would be truly, I think, revolutionary in the field. And I think in looking at physical, I actually think in dietary in the high-end nutritional epidemiology, there are models of how this is being done that actually the physical activity scientists could also take a look at, because they're so in love with their accelerometer. The accelerometer doesn't tell you what activity we're engaging in when you got your, such and such count. So from a translational point of view, we're missing something there too, right? So and I think the interest in measurement error correction is still not misplaced, especially if it becomes a little bit more accessible, because as I said before, even with 2 or 3 very good instruments, we will still have some error. Error is inherent in any data collection effort. So, and which is why other sciences call it noise and not error. I think error is also a very, judgmental term, if you will. So I think a little bit more openness on both of those ends would serve us well.
I just want to pick up on one subtle point you made there about, you mentioned that a lot of people think that more data brings us closer to the truth. And in statistics, we call this a big data paradox. And Xiaoli Meng coined that term. And actually, with more data, we get more precise estimates. But if people are underreporting, we get closer to an inaccurate answer. So it's very challenging. And I think that, as you mentioned, we think about longitudinal data collection. And, if we think about a time period like pregnancy, do we have consensus on the ideal times and best methodology for assessing a pregnant woman's intake? How many time points should we sample?
I may answer that. Hi, everybody. In pregnancy, this is a question that remains really important to know how it's better to assess dietary intakes. And I may answer a little bit of the question that was asked before. I think that if we, in the meantime, until we have the best method to assess dietary intakes, I think that we can work on what we have. And depending on what is the research question. If I'm interested in health outcomes or maternal or neonatal outcomes, and if my study demonstrated that the diet quality decreased from the beginning to the end of the pregnancy, I can start my intervention on that and say, okay, great, this is may not perfect, but I realize that fruit and vegetable decrease. So I can base my intervention on that. I think that maybe we don't have the perfect tool, but we have some information that may help intervention and that may help pregnant to eat better and to eat well. So to answer your question, Amy, I think that we need to assess more than one time during pregnancy. And I think that there are questions that remain. I think that we need to better understand energy intakes in association with current recommendations. Current recommendations are made for each trimester. So I think that we have to look what happened at each trimester. And how much more do pregnant women need to eat to have a healthy pregnant? Don't know. If we promote a quality diet with the right supplementation, is that enough to promote maternal health? Can we let physiology guide women's intakes? In my opinion, those questions remains unanswered. So I think the more we get information, the more we can work with pregnant women. But in the meantime, I think that health researchers can do something to help women to have a healthy pregnancy.
Thank you so much. And I just want to say a special thanks to all of our esteemed scientists and researchers who joined today and those of you who submitted questions. It's been a really interesting discussion. And with that, I would like to turn it back over to Dr. Cheryl Anderson.
Thank you.
Thank you so much, Amy, and thank you to everyone for a brilliant conversation this morning. I'm just going to go ahead and share my screen and try to do a very quick wrap up for us here today. So first, I want to just thank our speakers. You'll see that there we are. Thank you to our speakers and our panel discussants this morning. Quick summary. We gathered over the last 4 webinars to really understand pregnancy, a vulnerable time period where women are willing to make some dietary changes in ages 2 to 5. It's a time of life where our children are establishing preferences that can affect our health across the life course. And in 6 to 11, it's transitional. So there's some need to really think about multi-data collection activities and proxy reportings. We've got an evolving food market where our food, beverages, and supplements necessitate that we be flexible and really think about how we are approaching our data collection efforts. And we have these challenges as they came up again today with unobserved intake, waste, forgotten foods. How do we think about portion sizes? There's no ideal tool, right? So these emerging technologies and our innovations that we talked about today help us get a better handle on how do we match the tool to the question, how much burden is going to be tolerated, what level of accuracy we might get, the diversity concerns, literacy, what are we willing to tolerate, and how many resources do we have available to us? And don't forget our partners, right? We have been talking about how to increase precision in our assessment, and we're not the only nutrition scientists are not the only group that's working on these issues. So really think about bringing in engineers and other disciplines. As we are trying to get better at doing the assessment, things are changing rapidly, but we heard that tension around, what we can expect in the short term and what we might expect in the longer term. Perhaps, as was brought up this morning, at some point, self-report will be unacceptable. The multi-dimensionality framework can help us think about as we set patterns or expectations or recommendations for food and supplement intake. What about when people want to deviate from that slightly, right? How do we bring that into the research conversation? And then a big chunk of our conversation today was on harmonization. So whether it's about, supplement intake, which we have quite a bit of work to do on, or on a dietary assessment, we really will need user-friendly platforms that allow us, as we think across databases where we have repeated measures from various studies, to try and bring them in to fit into a sophisticated analytic methods. And then ultimately what we're trying to do is to share and integrate the data. We want to be able to answer our questions across well-conducted studies. And this is going to require us having an open source common data model that's put into a platform that our community can use. So we're going to need to standardize things. We need to share our data dictionaries, and we need to include any metadata that's going to make this work possible. So just 3 last comments. Ultimately, our goal is to determine bioavailable amounts of nutrients, right? You heard that commentary come forward. In each stage, that is what we want to do. So whether we're talking about our survey methods, our other, diaries, our biomarkers, they help us understand nutrient status, and we need to think in a way that is complementary, multidimensional. No tool is going to be perfect. I loved Dr. Herring's comment about noise rather than error, because it's going to be inherent to any data collection effort that we have. And so what we need to do is to really understand the lay of the land, really think about how to use these multi-modes and complementary tools in a way that serves our community well. So that is a largely, I'm sure, unsatisfactory summary of really amazing webinars that we've had. The recordings will be available shortly after this one closes. The others have been already posted. We'll make proceedings from the 4 webinars that will be available at the National Academies website. So with that, I just want to again say thank you to everyone for coming. And we had a fantastic discussion today. And in our other 3 webinars, please do go back and review them and share them with our colleagues. Take care, everybody, and look forward to the proceedings.